is essentially these little stop signs. You can't see those from the back. Those are stop signs. And basically what happens is that's where the checkpoints occur. They did it's kind of an eloquent little series of experiments that they talk about. That they got to find out what was happening. And essentially what happens here is that as a cell goes through each of the phases, G1 here, and there's a point, the S phase, G2, and then in mitosis, there's situations that can cause a stop. And if the stop happens, what happens is the cell goes into what's called a resting phase, and they represent that here as a GO phase, but I'm not going to ask you that. Point being is that things stop. And they stop so that the cell can repair itself and or not replicate errors. Okay, so the whole idea of the checkpoint system is to make sure things are done right, and if they're not, to stop and give the cell machinery time to go back and re-fix it, or just to stop it for good so that it doesn't continue on and propagate its own error. Okay, that's really it. All right, and there's different ones for. I'm not going to ask you where they are, any of that stuff. I just want you to get the idea that there are checkpoints and how that happens. Okay. Now, the kind of complex part about this whole thing is this, and this is where I told you don't get bogged down in the details because they had this huge detailed pathophysiology here. It mentioned the cyclins and how they all build up and break down and stuff. But what I really want you to get out of this is this. There are things called kinases, and guess what those are? ACEs. What's that suffix stand for? Enzymes, right on. So kinases are enzymes, and they talk about what the role they end up phosphorylating, ATP, and then they go ahead and take that phosphate and do everything. I don't care about that. It's too detailed for, as far as I'm concerned, for us. But the enzyme itself is pretty important because actually we were introduced to this kinase, different types of kinases, but they basically do the same thing back in cellular respiration. Back there, kinases played a role in the breakdown of glucose into glycolysis, and they played a role in mediating that through insulin and glycogen. So they play a similar function here, and again, they're always a very complex player in the game. Okay, and so what I want you to know is this. Cyclins are proteins that build up as they go through the cycle, and they get to a certain checkpoint. At that point, those cyclins are assessed, and everything is working. They are broken down, and then the system starts again, and they build up again, etc. Along the way, they will bind with an active form of kinase, okay, and activate it. And that kinase is what really does the work. That kinase will then continue the process or make changes, okay? So a complex interaction occurs between cyclins and kinases to regulate the checkpoints. Those are really the key players that will either stop or fix the errors as you go through the checkpoints, if that makes sense to you, okay? So know that these two are involved in the cell stopping or starting, and the fact that these two together, cyclins have to be around in order for kinases to be activated. And that's kind of a key little step there, and that's one of the things I want you to know. Yes, ma'am?
Going back to the graph on the previous slide, the G0,
yes.
What does that do?
That's when a cell is put on hold, or it hits a stop signal, it goes into a resting state. And at that point, it can stay there either indefinitely, and it'll eventually die, right? Or it will be fixed and continue. Okay, so but basically this is kind of like if it hits a stop sign, it's kind of like what would you call it? Like a detour, and it may stay there indefinitely until either it dies or it's fixed. Okay, so that's kind of just a bypass. It could occur here, could occur here, could occur here, could occur here. Okay, that's what it's. It's just a visual representation. Good question. All right, so again, key point here: kinases and cyclins are essential to for the checkpoints to be effective in the cell, and they then regulate overall whether or not the cell cycle continues or stops. Okay. All right. Now, we're going to talk about the normal situation, then we're going to talk about the abnormal situation. Okay. So to lay the groundwork, I want you to know what happens normally. In your textbook, there was a description of this. I'm going to show you the picture. Let me just go ahead right now to it, because this may look familiar to you. This picture, and it got very complex once again. And down on the bottom here, it had some labels of the DNA strand, and it used some terms: proto-oncogene, tumor suppressor gene, and then all the rest of the things, okay? So this is the normal situation, okay? So what I'm going to do is I'm going to describe this, and then I'm going to talk about what happens if something goes awry to cause this, which is cancer, okay? So let me back up. All right, so these things called proto-oncogene, that's a term you need to know. So if you don't have that down, proto, it's like before, an oncogene. An oncogene is a gene that causes cancer. Okay, onco is cancer, and a gene that causes cancer. Proto-oncogene is a gene that is before the gene that causes cancer. So it's really in a gene, okay? In normal scenarios, you have a start signal. And that start signal is what the proto-oncogenes typically are. Okay? They'll say start dividing. And that signal typically is sent from outside of a cell. And then there's another signal to stop division. And that's called the tumor suppressor gene. Tumor suppressor gene. Okay? And that's the stop signal. So this starts it, this stops it, and obviously those checkpoints play a role in whether